grade 12 learners. Welcome to today's Life Sciences lesson. My name is Ditoro Magwala, and this program is brought to you by the Northern Cape Department of Education in collaboration with Pagama Research and Development. So in today's lesson, we are looking at our meiosis revision, and we will be focusing on the process of meiosis, which means that we are going to be looking at the different phases of meiosis, both for Meiosis I and Meiosis II. So we are going to just have a revision of what happens during each phase, how you will be able to identify the phases by just looking at the diagrams provided. And also we are going to touch on a bit on the importance of meiosis. Right, so since this is your revision for your June examination, we are also just going to have a quick look at our question paper structure. So this is how the question paper will be structured. It is a paper out of 150 marks with section A and section B. For section A, we have 50 marks for our short questions. So here we will be looking at your multiple choice questions, terminologies, as well as the matching items question. Right, and then in section B, we have question 2 and question 3. Question 2 is 50 marks, and question 3 will also be 50 marks, and that will give us a total of 150 marks. One last reminder is that the paper is is written for 2.5 hours, which is 150 minutes. That means that you are only given 1 minute for 1 mark. So make sure that you make good use of your time. All right, so by the end of our lesson today, you should be able to list the 2 types of gametes. It is important for you to know the 2 gametes that we are going to be referring to. Remember, from our knowledge of reproduction, we know that the gametes are formed by meiosis, right? Reproduction and also genetics and inheritance. And then describe the human karyotype. You should know the human karyotype and know what makes up the karyotype. You should also be able to describe the number of chromosomes in somatic and sex cells. You should know the difference or to differentiate between haploid and diploid chromosomes, or diploid and haploid cells. You should also be able to describe the process of crossing over. And lastly, you should be able to describe the phases involved in the process of meiosis, which is what we are mainly going to be looking at here. All right, our pre-knowledge here would be the process of mitosis. We are going to look at how mitosis, we are just going to have a quick recap of mitosis as we are looking at meiosis. However, we know that just like meiosis, we have interphase all the way to telophase, right? Okay, so interphase, metaphase, prophase, metaphase, anaphase, and telophase, which also has cytokinesis, right? Okay, let's have a look at our baseline assessment quickly. All right, it says give the names of the parts labeled A to D. So this is just for you to get a good reminder of the structure of the chromosome, right? A, here we have a chromosome. B, we have the centromere. C, we have the chromatids. And lastly, we have homologous chromosomes, or homologous pair of chromosomes. Okay, so this means that you should be able to know the difference between a single chromosome and a homologous pair of chromosomes. When there's 2 of them like this, they are referred to as a homologous pair of chromosomes. And when you're only looking at 1 chromosome, you are looking at just a chromosome, right? And these 2 parts that make up this chromosome are referred to as chromatids, and they are held together by the centromere, right? Okay, so there we have our answers: the chromosome, centromere, chromatids, and homologous chromosomes. Right, so as I said earlier, you should be able to know what kind of chromosomes do we find in a somatic cell and what kind of chromosomes do we find in gametes. By gametes, remember, we are referring to the sex cells. So sex cells are also called gametes, right? So we have somatic cells, which are our body cells. These body cells are found throughout the body, and they are as a result of mitosis. Remember, and then we have the sex cells, or these gametes, which are as a result of meiosis, which is what we are going to be looking at in today's lesson. right? So these body cells, which are also known as somatic cells, are the cells that we find throughout the body. So in your skin, in your lungs, in your heart, in your liver, all over your body, you are going to find these body cells or these somatic cells. And another thing that you should remember about somatic cells is that because they are made or produced via mitosis, it then means that they are identical. So the cells of your body are all identical, excluding these cells that were made from meiosis, which are your gametes. So these ones are genetically different from each other and from the parent cell that they arose from. So here, this would be an example of our somatic cells. And as we can see, the gametes are haploid because they're made up of only one chromosome or one set of chromosomes, right? Whereas the chromosomes that we are seeing in the somatic cells are diploid. Another thing you should also remember about mitosis and meiosis is that mitosis produces cells that are not haploid. The cells are going to be exactly the same as the original cell, whereas one of the main aims of meiosis is to produce cells that are haploid. Hence, we are only seeing one set of chromosomes in that gamete cell. Right. Okay, as I just said, somatic cells are always diploid, and the chromosomes are always found in pairs, right? And we know that in humans, because we have 46 chromosomes, it means we have 23 pairs of these chromosomes, right? And it is because half the number of chromosomes were inherited from the mother and half the number of chromosomes were inherited from the father, right? So half is from the maternal origin and half is from the paternal origin, resulting in a diploid individual or an individual that contains diploid cells. Okay, so our gametes or our sex cells, here we are referring to the sperm cell and the ovum in females and the sperm cells in males, right? So those are our two types of gametes, and both these gametes are haploid. They contain 23 single chromosomes. With the sperm cells and the ovum, we say they are haploid, and haploid is represented by the letter N. Whereas with the somatic cells, we say they are diploid, and that would be represented by the number 2n or the letter 2n, right? So 2n means diploid, n means haploid, right? And with the gametes, they were produced by meiosis, and the somatic cells would be produced by the process of mitosis, right? So as I just said, haploid is represented by the symbol N, and 23 is that number of chromosomes in humans. And then diploid would then be represented by 2n. Diploid would be 2n, and it is 46 chromosomes in humans. We know that 23 is half of 46, so that would make sense. Right, now let's have a look at the phases of meiosis. Right, if we have a look at this diagram over here, it is just giving us an overview of the whole process of meiosis, starting from interphase, which is the phase that prepares the cell for either mitosis or meiosis. Here we are looking at meiosis, so this is in preparation of the actual process of meiosis. And then we can see here at the beginning, at the end of meiosis 1, this is two cells that we would have ended up with that are haploid. And then at the end of meiosis 2, again here, we can see that now we have 4 cells that are genetically different from each other. So if we have a look at this, we would see that we started off with a cell that has 4 chromosomes. And then we ended up with 4 cells that have 2 chromosomes. Can you see that at the end of meiosis, these cells have now halved the chromosome numbers from 4 to 2, meaning that they have halved the chromosome numbers from 2n to n, right? Because as we have just said, meiosis produces cells that are haploid. Okay, this process, as I just said here, meiosis 1 and meiosis 2, it occurs in 2 phases, which are meiosis 1 and meiosis 2, and we are going to look at exactly what happens during these phases. And we are also going to look at each phase and see how it differs with the other one. So, which means that we're going to look at meiosis 1 and look at what happens during prophase 1 of meiosis 1 and what happens during prophase 2 of meiosis 2. Right, okay, and then each of the cells that are produced by meiosis 1 are going to be genetically different from each other. They're going to have genetically different material. And during meiosis 2, the cells that were produced at meiosis 1 are now going to undergo meiosis 2 separately, and they're each going to produce 2 cells. As you can see, these 2 cells are from this one cell, and these 2 cells are from this one cell, resulting into 4 cells. Right, 4 cells that are genetically different from each other. Now we are going to get a reminder of why these cells end up being genetically different from each other at the end of the whole process. Okay, starting off with meiosis 1, we know that here we start off by looking at interphase. Interphase is a preparation phase for meiosis 1. And during this process, we know that that is when DNA replication takes place. The DNA replicates, the genetic material becomes replicated as well. Right, so the chromatin network is going to be doubled, or the genetic material in the chromatin network is going to be doubled during this phase. Right, so this is basically what happens during interphase. The main thing that happens there is DNA replication. Okay, next we have prophase. Also, it is very important for you to know the order, because we start off with interphase, prophase, metaphase, anaphase, and telophase. So you should remind yourself, always remind yourself the order of how these phases occur, because it is no use understanding the phases but not knowing the correct order. Right. Now, also looking at the diagram here, as we are looking at the points, we are also going to be looking at the diagram. Firstly, the nuclear membrane is going to start to disappear. Here is the nuclear membrane. It's looking like dotted lines because it is starting to disappear. The nucleolus also starts to disappear. This nucleolus is starting to disappear as well. The chromatin network is going to unwind, and we can see that now chromosomes are becoming visible. We can really see that there are chromosomes that are visible here. The next point, and one of the most important things about prophase 1, not just prophase, but prophase 1, one of the most important things here is the fact that crossing over takes place. Right, so when crossing over takes place, we are going to see that the genetic material between the 2 chromosomes is going to be exchanged, and when this exchanges, it leads to the chromosomes not being the same as they were at the beginning. So this is where the start of cells being genetically different starts. Cells become genetically different because of this process of crossing over. Okay, let's have a look at that. So the 2 chromosomes, the homologous pair of chromosomes. This chromosome is from the father and this chromosome is from the mother. Right? So these 2 chromosomes are going to lie next to each other. As they lie next to each other like this, we refer to them as a homologous pair of chromosomes or homologous chromosomes. Right, so this homologous chromosome, as they lie next to each other like this, they are going to have the chromatids. Each of the chromatids from each parent chromosome is going to overlap with the chromatid of the other chromosome. So this chromatid is going to overlap with this chromatid, right? Once that happens, it means that they're going to exchange genetic material and then they're going to separate from each other, like what is happening here. So they're going to separate. At that process of separating, the one chromosome is going to remain with the genetic material of the other chromosome, and the other one is also going to remain with the genetic material of its homologous chromosome. And the point where this happens, the point of crossing over, this part is called chiasma, and then in plural is called chiasmata. Now, one chromatid of each chromosome overlaps with the chromatid of the other chromosome. That is what I've just mentioned. The point of crossing over is called chiasmata or chiasma. Okay, and then the chromosomes are going to separate. As they are separating, we can see here they have already separated, and each chromosome will now contain genetic material from the other chromosome. We can see here by this color coding that the red chromosome has been now contains part of the blue chromosome, and the blue chromosome also now contains part of the red chromosome. So that means that the genetic material from the blue chromosome is now part of the red one, and vice versa. We can see this here because originally we had capital letter A, B, and C. But now we will see that we have A, B, and a small letter C. So that is one of the ways in which we can see that genetic material here has been exchanged. Right, and then from the diagram we can see that the chromosome is made up of one original chromatid. So this is the original chromatid with genetic material from the homologue partner, and then we have the other chromosome with genetic material from the homologue partner. So now whatever happens as the chromosomes are moving on within the process of meiosis with the other phases, these two chromosomes or these two chromatids are now no longer the same with each other. These two chromatids are now different from each other. Same applies to these chromatids. They are now different from each other, meaning that that process of making sure that the cells at the end of meiosis are different is getting there, because we already have chromosomes that are different. No matter what happens, the chromosomes are different from each other. Right. Why is it important for this crossing over to take place? Right. So it results in the exchange of genetic material, which is what we just looked at, leading to cells that are genetically different from each other and from the parent. Okay. And this means that the gametes will be different from each other. The gametes formed will be different from each other. Remember, here when we are talking about the gametes, we're talking about the sperm cells and the ovum. Now, we have siblings from the same parents, and they do not look alike at all, or they may slightly look alike, but they have differences here and there, behavioral differences, and all of these. That is because the gametes that they were produced from were different. So each time a male produces sperm cells, those sperm cells are going to be different from each other. Each time a female produces an ovum, that ovum is going to be different from the previous ovum. So that allows us to produce offspring that are genetically different from each other, which at the end of the day increases genetic variation, which is one concept that is very important. It is important for us to have organisms that are genetically different from each other. However, we will focus on that a bit more later. All right, now let's move on to metaphase. Right, so now here as we look at this, we will see that the chromosomes are aligned at the equator in their homologous pairs. Right, we can also see that the chromosomes that have went through crossing over are now containing that the different genetic material from the homologous chromosome. Right, so here chromosomes align at the equator in homologous pairs. Homologous pairs of chromosomes align along the center of the cell. This is a very important statement about the process of metaphase, or the phase or metaphase, right? Because that is the main thing that happens here. Now, another thing that you need to remember about these chromosomes aligning at the equator is that they are going to align at the equator randomly. So as much as they are aligning here in homologous pairs, they are going to be aligning randomly. As you can see here, we have the green one on our left, and we have the blue one on our right here. And here we have the blue one on our left and the green one on our right. It doesn't mean this is how it's always going to be. Sometimes you're going to find a few blue ones on the left and a few green ones on the right, and vice versa. There isn't even a set number of how many of the maternal origin should be on which side and how many of the paternal origin should be on the other side. So they're just going to align there randomly. So right? Remember, this is just us using 4 chromosomes as an example, but in real life sense, in humans, this will be happening with 46 chromosomes or 23 pairs of chromosomes, right? So they're going to lie at the equator in homologous pairs, and that will be randomly so. So the random arrangement of chromosomes at the equator of the cell during metaphase. This allows for the cells produced to be genetically different once more, right? So it just increases to that genetic variation that we spoke about. Okay, and then we have anaphase. Anaphase is when the spindle fibers over here, these are the spindle fibers, remember? When these spindle fibers are going to shorten. So when the spindle fibers shorten or contract, it means that they are going to pull on these chromosomes as well. They're going to pull the chromosomes to the opposite sides. Right, when they pull these chromosomes to the opposite sides like that, they're going to pull them in the order that they were aligned at the equator. So if the blue ones were on the left and the green ones were on the right, it means that now all the ones that were on the left will go to the far left, to the poles of the left side, and the ones that were on the right will also go to the poles of the right side, which is now taking us towards the division of the cytoplasm, whereby we are going to now end up with two cells that are produced at the end of meiosis I. So here the spindle fiber will contract, and it is going to pull the chromosomes apart as it shortens, because the chromosomes are attached to the spindle fibers. And then whole chromosomes are going to move to the opposite poles. Here we are looking at whole chromosomes moving to the opposite poles. It is still a chromosome, it's still a chromosome like that, meaning that we are not looking at chromatids being pulled to the opposite side. Remember, we know that during anaphase 2, it is going to be the chromatids that are pulled to the opposite side and not the whole chromosomes. So as we are going on like this should just be a reminder for you that whatever happens in meiosis I is not necessarily the same as meiosis II. Okay, the last phase here, we are looking at telophase 1. Here now we have a group of chromosomes on each pole. We have the chromosomes on the left and the chromosomes are on the right. Okay, the nuclear membrane is going to start to show up again. It's going to reform, and then the cytoplasm will start to divide, right? Once the cytoplasm divides, it means that we are going to end up with two cells. We're going to end up two cells, which each have two chromosomes. We started off with 4 chromosomes in the cell. Now we have 2 cells with 2 chromosomes each. It means that the 2 cells have shared this genetic material with each other, and now the chromosome numbers within a cell have already been halved. Okay, and then there is going to be a formation of the nucleus at each pole. Okay, cytoplasm divides and it's going to form a cleavage furrow. When this happens, we are looking at cytokinesis. Okay, which is what we are looking at now here. So this is the division of the cytoplasm, cytokinesis, and then we are going to see the 2 cells that have resulted in this being formed right here at the bottom. Okay, 2 cells will result. This will result in 2 cells. Each new cell has half the number of chromosomes. As we can see in the previous diagrams, we can see that we started off with a cell that had 4 chromosomes. Now we are ending up with 2 cells which have half the number of chromosomes. So we started off with a 2N cell, and now we are ending up with 1N cell and another N cell, which means haploid and haploid will give us diploid. Okay, now this is how we would have cytokinesis taking place with the 2 cells that have the number of chromosomes being halved, which means that we have N over here. So the cells are different from each other and the original cell. They have genetic material that is different from each other. So that is due to the random arrangement of chromosomes at the center of the cell at the equator, and it is also due to crossing over during prophase 1. So crossing over and the random arrangement of chromosomes leads to cells that are genetically different from each other. These cells are different from each other. They do not look alike, and they do not have the same number of chromosomes as the parent cell. They have haploid chromosomes. Okay, and then here we have activity 1. It says the diagram shows a process during meiosis. The letters represent the alleles of genes in the chromosomes. So we have here part A, chromatid, B, a chromosome, and then C, the centromere, and then D is chiasma, right? And then as you can see here, we have RT and D, the way it is represented here, and then the same applies here. And then on here we have capital R, small t, capital N, small n and capital D. So now, before we can even move on to the questions, we can see that here crossing over is taking place, meaning that this black chromosome will now end up with RT and the white part ND, right? And then the white chromosome is going to end up with RT and D. No, and D, like that, with a small letter D. So it will be RT N D. And then the other one is the opposite, small letter N and capital letter D. Here, capital letter N, small letter D. They have exchanged genetic material now, right? Okay, let's look at the questions. We have 1.4.1 up to 1.4.4. Name the process shown in the diagram. So we can see that the process that is taking place there is crossing over. Make sure, guys, that you do not confuse a process and a phase. When the question says name the process, you name the process. This is the random arrangement of chromosomes. This is crossing over, right? Whereas if they say name the phase, you can either say interphase, prophase, metaphase, anaphase, any of those phases. But when they talk about the diagram or the process, they are meaning you should name what process or what event is taking place there. So we can see that it is crossing over. And then B, the phase of meiosis represented above. When does crossing over take place? Crossing over takes place during prophase. So the answer for B there would be prophase 1. Different from prophase 2, therefore, this only takes place in prophase 1. Okay, not in prophase 2. So you must make sure that you say prophase 1 when you answer the question, because once you say prophase only, we don't know whether you mean prophase 1 or prophase 2, so that becomes a problem. 1.4.2, identify point D. Let's have a look at what point D is. That is chiasma. So that is the point where the chromosomes are going to overlap with each other. Okay, and then part B. Let's have a look at part B once more. There we have part B. That is a chromosome. It is not a homologous pair of chromosomes, but it is just a chromosome. And then part A, we can see that that is a chromatid. Okay, let's move on to 1.4.3. Write down the letter and name of the part that join together two chromatids. The part that joins two chromatids together is called the centromere, right? And that is part C on our diagram. So part C at the center there is referred to as the centromere. 1.4.4, write down the genotypes of the resulting chromatid A after the process named in question 1.4.1. So after crossing over, what is going to happen to the genetic material of those chromosomes? So that is actually needed here. So as I said, for this chromosome at B, which is the white one, it is now going to have capital letter R, small letter T, and capital N, small letter D. This is the new one, right? And then the blue, the green, the black one, we'll have small letter R, capital T, and small letter N, capital D, like that. Okay, so that is what will be happening there, whereby we are looking at chromatid A. So let's have a look at chromatid A specifically. This is chromatid A, which is the black one. Therefore, it's small letter R, capital T, right? Small letter N, capital D, because it's taking this part of the white chromosome. So it is the small letter R, capital letter D part. So that is very important for you to understand that as these cells undergo crossing over, they are going to exchange genetic material. And this will be from the point of chiasma, whereby they have overlapped and exchanged genetic material. So this is how it would look like. Okay, moving right along to meiosis II. With meiosis II, remember, we do not have interphase taking place here. There is no interphase, there is no DNA replication. We go straight to prophase II. Main thing you should know here is that crossing over is not taking place. The chromosomes or the chromatids are not going to exchange genetic material. Therefore, they are not going to become more and more different from each other. Now, remember, at the end of meiosis I, two cells were produced. What happens to those two cells? They are now going to be part of meiosis II. They are now going to undergo meiosis II. As they are undergoing meiosis II, we will see that they are going to divide at the end, and they are going to have those gametes being produced, or those haploid cells being produced at the end. Right, here we can see the distinct chromosomes at prophase II, and we can see that there are two chromosomes that are visible, chromatids that are visible per chromosome, as you can see, right? And then we will see that the centrioles will also move to the opposite poles. So these centrioles are going to move to the opposite poles, as you can see, and the spindle fibers are going to appear. Right, nuclear membrane again, just like prophase I, the nuclear membrane is going to disappear, as well as the nucleolus, they are going to disappear. No crossing over in this case during prophase II. Right, in prophase, in metaphase II now, chromosomes again are aligned at the equator, randomly so. But the difference is that they are no longer aligned in homologous pairs. Remember, with what we were looking at, they were aligned like this in homologous pairs in metaphase I. Now in metaphase II, they are aligned singly. Chromosomes are aligned singly at the equator of the cell. Chromosomes align along the center of the cell. So these chromosomes are not in homologous pairs, but they are just single. Remember, they align randomly. Right, so the spindle fibers also are attaching themselves to the centromere of the chromosomes. So the chromosomes are held there with the spindle fibers right there at the center. Okay, we can see that. And then anaphase, we know during anaphase we focus on the chromosomes separating. We focus on the separation of the chromosomes from each other by the shortening of the spindle fibers. As the spindle fibers separate or shorten, they're going to pull the chromatids of each chromosome apart from each other. So you can see here, this is the chromosome, the chromatid, and this is another chromatid. They were attached to each other at the center. But now when the spindle fibers shortened, they pulled the chromosomes apart. Right? Also on this side, they pulled the chromosomes apart, and each chromatid now was pulled to the opposite poles. Right. Here again, very important for you to mention that it is not whole chromosomes that were pulled to the opposite poles, but it was the chromatids. Now, when these chromatids are not part of a chromosome, remember we have chromatids here. This is a chromatid, another chromatid. They form a chromosome together with the centromere. Now, if it's just a chromatid like this on its own, we do not refer to that as a chromatid, but we refer to that as an unreplicated chromosome. Very important also for you to remember. When you count the number of chromosomes at the end of meiosis, you do not say, but this is not a chromosome, this is a chromatid. How am I counting chromosomes at the end of meiosis when I only have chromatids? They are referred to unreplicated chromosomes, therefore they are still chromosomes. Right, so don't make that mistake of saying, but this is not a chromosome, this is a chromatid. It is a chromosome only when they are attached. When they are just chromatids like that, we refer to them as unreplicated chromosomes. Right, so that is how we are going to see this. They are going to actually be just single like that. Now, they are also going to be genetically different from each other. They are going to be different from each other because of the random arrangement that happened, and also for the crossing over, because of the crossing over that took place. Now, how does this become possible for the chromosomes to be still the same as what happened during meiosis I? Now the chromosome numbers have not halved again. If you started off with 4 chromosomes at the beginning, they undergo meiosis I and you end up with 2 chromosomes in a cell, and they undergo meiosis II and you still end up with 2 chromosomes. How was that possible? Why do we not now end up with 1? Because it has halved here to 2. Why is it not halving to 1 now? This is because of the chromosomes now being split in the middle into chromatids, which we can now refer to as unreplicated chromosomes. This is because of that process of DNA replication during interphase. The DNA was doubled, the DNA was replicated. So we still have genetic material, we still have these chromosomes, just that they are unreplicated, and they can replicate again in order for the genetic material to double once more. All right, and then the last part, which is telophase 2. Now we can see here the cytoplasm is separating, the chromosomes are moving to the far end of the poles, and we can see that we are starting to see 2 cells resulting from each cell that was at the beginning of meiosis II. So this was 1 cell, now it is producing 2 cells. This was another cell, it is also producing 2 cells. Now these 2 cells, these 4 cells are going to be what we see at the end of meiosis. Meiosis produces 4 cells that are genetically different from each other. Totally genetic from each other. What causes this genetic difference? Crossing over. When does crossing over take place? Prophase. Very important. These are some of those important questions that you will be asked when you are doing meiosis. What else is causing them to be genetically different? It's random arrangement of chromosomes at the equator. Random. Them being randomly arranged is very important, because if they are not arranged randomly, the cells will not be too different from each other. Random arrangement of chromosomes at the equator. When does this random arrangement take place? Random arrangement takes place during metaphase 1 and 2, whereas prophase 1 is the only phase that has this process of crossing over. There is no crossing over during prophase 2. Okay, there are now 2 groups of chromosomes at each end of the pole. As we can see there, we have them, and then you can see the chromosome numbers are still 2. Then they have not halved to 1, so they remain 2. There is no halving effect during meiosis II, right? These groups of chromosomes will now go on to be part of the new cell, right? Okay, so we have the new nucleus within that cell, and the cytoplasm will also divide to form 2 cells with half the genetic material each. 2 cells with genetic material that is different, causing those cells to be genetically different from each other, and therefore causing all the cells that have been produced here to be different from each other. So now the 2 cells have undergone meiosis II and have produced 2 cells each, meaning that they are now 4 cells. Each of these cells genetically different from each other, and they are also different from the original cell. They also contain half the genetic material, the number of chromosomes. We started off with 4, we ended up with 2. If you start off with 46 in humans, you are going to end up with 23 chromosomes, and that is the characteristic that we look at when we look at the number of chromosomes in a gamete. A gamete must be haploid. A gamete must contain 23 chromosomes and not 46. It should be haploid. Okay, let's have a look at activity 2. The diagram shows a phase of meiosis. Okay, before you try to now move on to the questions, you label whatever labels you are given, and you also try to identify which phase am I looking at. So from looking at this, we can see the spindle fibers here attaching to the central we can see that chromosomes are still in homologous pairs. Once chromosomes are in homologous pairs, you know that this is meiosis I. That is a definite, because in meiosis II, chromosomes are now single chromosomes, right? And then if the spindle fibers are attached like this and the chromosomes are at the center of the cell, it means that we are looking at metaphase I. Right, so by the time you get to the questions and the question is asking which phase, you know. This one here is the centromere, and this is a homologous pair of chromosomes. Okay, 2.3.1 up to 2.3.4. It says here, name the parts labeled X and Y. So as we can see, X is the centromere and Y is the homologous chromosomes. Centromere and homologous chromosomes. Name the phase that is shown. We have already established that that is metaphase I. If you do not specify that it is metaphase I, you are not going to get that mark, because it could be then metaphase II. Make sure you specify. Describe the chromosomal behavior named in question 2.3.2. So what happens to the chromosomes during metaphase I? Chromosomes are going to align at the equator, in homologous pairs, and they are going to align there randomly. So they align at the equator in homologous pairs, and they align randomly. Okay, we are going to look at the answers just now. And lastly, explain one importance of the chromosomal behavior in 2.3.3. Why is it important for the chromosomes to align at the equator? And why is it important for them to align there randomly? They align at the equator so that they can be pulled to the opposite poles. And when this happens, the cells that are going to result are going to be further different from each other. So when they align at the equator in homologous pairs, each chromosome for each pair is going to now be pulled to each pole in order to form those two cells at the end of meiosis I. Okay, so there we have our centromere and homologous chromosomes. We have metaphase, and then the behavior, what happens during metaphase I. Homologous chromosomes are randomly arranged at the equator, and they are attached to spindle fibers. So any of these can give you those 3 marks that were asked. So you should be able to give all of this. Chromosomes randomly arranged at the equator, and they are attached to spindle fibers. You don't just say chromosomes, you say homologous chromosomes. If you do not say homologous, it means you are talking about metaphase II. So make sure that you mention that it is homologous chromosomes. This one we said, why is it important for this random arrangement of chromosomes to take place at the equator in metaphase I? Let's have a look at the 3 options that we have. The random arrangement of chromosomes introduce genetic variation. When the chromosomes are randomly arranged, it means that the cells produced are going to be different, which is mainly why we want meiosis to take place, for the cells to be different from each other. When cells are different from each other, we say there is genetic variation. The offsprings that we are going to produce are going to be different from each other. This may lead to organisms which are better suited to a changing environment. When the environment changes and the organisms produced are different from each other, it means that some are going to be well adapted to that environment, they can survive better, whereas some are not going to be well adapted and they will not survive. Unlike when all the chromosomes are the same, all the cells are the same, all the offspring produced are the same, and therefore when there is a changing environment, all the organisms produced from the same cells will then disappear, and that could lead to the extinction of a species, right? So we need genetic variation so that organisms can easily survive. Okay, another option, it results in chromosomes moving apart. When the chromosomes are attached to the spindle fibers, when the spindle fibers contract, they can pull them to the opposite poles, and that leads to the halving of the chromosome numbers. Another reason why meiosis is important, it has the halving effect. We want the chromosome numbers to be halved. We know that this is important for fertilization. During fertilization, when the haploid sperm cell and the haploid ovum come together, resulting in a diploid ovum. So we need the gametes to be haploid. So metaphase leads to that halving effect, metaphase 1 and metaphase 2. The contraction of the spindle fibers allows the chromosomes to separate. When the chromosomes separate again, we are looking at the fact that the cells produced will now be haploid. Okay, let's quickly just summarize what we just looked at. Mainly here we were looking at Meiosis I and Meiosis II. What were we focusing on? The understanding of what happens during each phase and why these events were taking place during each phase. Each phase has its own event that is very important for the whole process, starting with interphase. Here we look at DNA replication. For the cells to have the number of chromosomes that they have at the end, it is due to the fact that DNA replication took place at the beginning. Right, prophase, crossing over. We need the cells to be genetically different from each other, therefore crossing over is very important. Metaphase, metaphase 1, random arrangement of chromosomes at the equator. Random arrangement. We just looked at this with activity 2, whereby we looked at why is it important for the random arrangement to take place and why is it important for them to be attached to the spindle fibers, whereby they will be pulled to the opposite poles. Anaphase 1, they are pulled to the opposite poles, allowing them to now form two separate groups, and that will allow for the formation of two new cells. Okay, telophase, telophase 1. Right, here with telophase 1, the cells are now separating and we are ending up with two cells that are now haploid. They are now haploid because they have been pulled apart, right? So that is now showing us why it was important for anaphase to take place. Moving on to Meiosis II, Prophase 2. Here we do not experience the crossing over, but we can see that there is the spindle fibers forming, and at the end they will end up attaching to the chromosomes, which is important because we need them to split the chromosomes into chromatids during anaphase 2. Metaphase 2, again, we have the random arrangement of chromosomes. However, in this case, they are in singles, they are not in homologous pairs. Something for you to remember. Metaphase 1, they are arranged in homologous pairs. Metaphase 2, they are arranged singly. Anaphase, here chromatids are being pulled to the opposite poles. Anaphase 1, whole chromosomes are being pulled to the opposite poles. Make sure that you do not miss that. Again, if you say anaphase and you don't say anaphase 1, it means that you don't know the difference between what happens during anaphase 1 and anaphase 2. Now make sure that you do not miss that. You always make sure that you give your answers accordingly in terms of which phase, which anaphase is this, or which metaphase or telophase is this. Telophase 2, now we can see that we are getting to the end and the results that we expected are starting to show. We started off with one cell, and now we are starting to see that we have the formation of cells that are 4 cells being haploid as well, whereas we started off with a diploid cell. So we can see the chromosomes are at the opposite poles, new cells are forming, the nuclei are forming, And now we can see that we have those unreplicated chromosomes. And you don't even have to say they are unreplicated. However, it is important for you to understand why we are referring to them as unreplicated chromosomes instead of chromatids. Because now we cannot say cells have been produced and they have half the number of chromosomes if we are referring to chromatids. In this case, they are the unreplicated chromosomes which have the potential to replicate. Right. And then at the end of our cytokinesis, we can see there, we now have four haploid cells, four cells that are haploid, four cells that have half the number of chromosomes, and cells that are genetically different from each other. We know they are different from each other because of crossing over in prophase 1. They are different from each other because of the random arrangement of chromosomes in both metaphase 1 and metaphase 2. Okay, now for your homework, here you are going to look at another phase whereby we can see that crossing over has already taken place. You have part A, B, C, and D, and then you will answer the questions that have been provided below, and that will be discussed in your next lesson. All right, let's have a look at the preview of our next lesson. So in our next lesson, we will be looking at the consequences of abnormal meiosis. So as we were discussing meiosis now, we were looking at it in the form that it should happen when everything is going well, when everything is taking place normally. However, we need to remember that some mistakes can happen, some errors may happen. Now, when these errors happen, what is the result of that? What are the consequences of that? So we're going to look at the consequences of abnormal meiosis, what happens if meiosis can take place abnormally, and the results of these abnormalities. So we are going to look at things such as Down syndrome, which can result if there are some errors that can happen during meiosis. All right, we have come to the end of our lesson for today. Thank you so much for watching. I will see you next time. Goodbye.